Nicole?
So I'm Nicole Gormley. I am the acting deputy division director in the division of heme products, where we review both benign and malignant products for those indications. And so again, I'll make the qualifier that these comments that I'm going to share with you are just my own perspective and do not necessarily represent. Oh, sure. These comments are those of my own personal experience. And I would also, just highlight that a lot of my examples, again, will come from this disease space of, malignant hematology, where we're dealing with, life-threatening conditions. And so, for us to really evaluate a drug and ultimately approve it, there's a requirement that there's demonstration of substantial evidence of efficacy with adequate demonstration of safety to enable the safe and effective use of the product. And ultimately, there must be enough information contained in the application that allows us to generate a product label that defines a specific patient population and also provide enough information in the label to allow for its safe and effective use. And so with the Cures Act, there's this focus now on the use of real-world data and evidence to really sort of expand the use and expedite drug development, et cetera. But I think just one of the important items to underscore is that the evidentiary criteria and standard really doesn't change, and that there still must be evidence of substantial evidence of efficacy and adequate safety demonstration. And, really in these settings, real-world data could be used in certain settings as the primary evidence, or it could be supportive evidence when considered in the totality of evidence that one considers when evaluating a product for approval. But there's certain specific, aspects that we as real-world data consumers and regulators would look at specifically when evaluating any data. And the first, really pertains to the patient population and the setting in which it's being used. And so there's several examples that come to mind, and this is also true when we're looking at randomized controlled data, that ultimately, we're looking at the data and saying, how well does this represent the proposed indication that's being put forth? And so, if we're looking at a product with a randomized controlled data for a patient population with AML that's been deemed unfit for intensive chemotherapy, with a randomized trial, we have, strict inclusion and exclusion criteria that would outline, the frailty criteria or other comorbid conditions that would define a patient as being unfit for intensive chemotherapy. And if we're looking at real-world data, that may just be identified by patients with a new diagnosis of AML that then go on to receive azacitidine or low-dose Ara-C. And so we'd be looking, at a really sort of a population defined by the treatment they receive. And there's the potential in some cases for bias to be introduced there, just in that there may be multiple, factors that contribute to a patient receiving a particular treatment. And so I think one aspect is you really have to have confidence when you're looking at the data that the patient population ultimately represents the indication that's being sought, and that there's enough information to have confidence in that population and then label it appropriately. Another aspect of this is the outcome that's being measured, and in some disease settings, and this is easier with some endpoints than with others. Obviously, overall survival in both randomized controlled trials and in real-world data is relatively easy to assess. If you're looking at response rate, it gets a little bit more challenging, and some endpoints might be really suitable to real-world data collection, et cetera, but still have some unique challenges. For example, if we're considering MDS and an endpoint of attaining transfusion independence, in theory, that could be very easily assessed with real-world data, just the presence or absence of transfusions. However, you have to have confidence that you're actually fully capturing the endpoint or the outcome measure. If patients receive transfusions at other centers, or for some odd reason that the data may not be complete, it may be difficult to have confidence in that outcome measurement. And so another aspect is just, ensuring that the reliability of the data and the methodologies used to collect that information. And as we've had discussions earlier today and yesterday, it's important to have confidence that the protocol is designed in such a way to minimize bias and account for any potential known or unknown confounders. The data capture should be complete, and there are methods that can allow us to have more confidence in the data that has been collected. Additionally, including, and we've heard some examples of this, combining data from different sources. So if we have data from primarily from claims databases, is it possible to combine that with electronic health records data? Various methodologies to sort of enhance or strengthen the confidence that we have. And specifically, looking to make sure that we have unique patients identified in the data sets. And then, as has been discussed as well, how much of this population with the data set then that we ultimately do have, how is representative of this, the general population that the indication is being sought? How representative is that the data set of that entire population? So there are another discussion just that we've had ultimately the statistical analyses that are applied should be robust and of significant rigor such that we can have confidence in the data, and the amount of missing data in general should be minimized, and there are methods to sort of address this. And I think, just in general, from the perspective, I think, the real-world evidence really has a significant potential to really expedite drug development and also to complement the data that we have currently available to us from randomized controlled trials. It's just that, it's important to make sure that we are, conscious of these potential confounders and biases and minimize them whenever possible. And from the agency perspective, I think I'll echo some comments that were made earlier by Dr. Martin. And it's just that it's exquisitely important, I think, to engage with us early in the process. We are, I think, very encouraged and hopeful that this really can be very helpful. But I think it's important that we have a dialogue throughout the process with specific sponsors considering using this method. And with that, I'll conclude. Thank you very much.
Great. Thanks, Nicole.